Hello everybody, welcome to NEET Simplified, your trusted space for simple explanations of complicated concepts. So here I bring another episode today for you to understand about the sexual reproduction in flowering plants, and we are beginning this chapter with the microsporogenesis and pollen development. Before we go there, do let me know in the comments from where are you watching this video from, so that we understand how many NEET champions are watching us right now. So to begin with, let's understand the structure of stamen, and then we'll go ahead for the microsporogenesis and the pollen development. Coming to the structure of stamen, we are all aware of the simple structure of stamen, which has a filament and the anther. Let's understand that we find two types of stamens. One are free and the other are epipetalis. Free in the sense they are only attached to the thalamus, which is nothing but the seat of the floral organs. So such stamens we call these as free stamens, whereas few you will find them attached to the petals, we call them as epipetalis stamens. And now the distal end of the stamen is that which is away from the plant body, and proximal end is that which is close to the plant body or close to the thalamus. Now, as you can see the microscope image, and as we always tell you, this is the only NEET zone where we get you the images captured through the microscope, so that you get each and every detail of what you are studying in your textbook through the diagrams. So this is how the anther looks under the microscope, with a longitudinal groove running lengthwise, and the outline of this anther is going to be tetragonal in structure. So as you can see, this is our tetragonal structure looks like. Let us understand in detail about the morphology of anther. Now this is what you see, this is the aerial view of the anther. You have found in your textbooks that anthers are bilobed, dithecous, tetrasporangiate. Now these are some terms which are really difficult to remember, right? So let's correlate this with a simple example so that you can relate and remember. Now let us consider very beautiful insect which we find regularly around us, the butterfly. Now coming to the butterfly, let us consider that half of its body with two of its wings forming one lobe. So this is lobe number 1. Now you have A and B, two wings here. Similarly, C and D are other two wings. So you have lobe number 1 here and lobe number 2 over here. So we have lobe 2 over here. So let us correlate this with the anther. Just like how you have find lobe number 1, this is lobe 1 with A and B. The wings you have seen here, isn't it? Just like that, A and B, these are the microsporangia which you find. And let us see the lobe number 2 as well. So this is lobe number 2 with C and D microsporangia. Now what are we doing? We are comparing the lobe to half of the butterfly's body with the two wings. So that is forming one lobe. Just like that, you have the wings corresponding to the microsporangia. So how many wings did we find? We found A, B, C and D, that is 4 wings. So just like the butterfly, we have 4 sporangia, hence tetrasporangiate. Also, we have found how many lobes? This is forming one unit. So this is the lobe number 1 and lobe number 2. So it is bilobed. Now you have also understood that there was one more term, dithecous. So now it's clear that there are 2 lobes and 4 microsporangia. Now what about theca? Yes, now let's very simply understand that A and B, these are the 2 wings which you are seeing here. These are on the same side. Similarly, A and B, these are the sporangia which are originated together. They share a common origin. Also, they are going to be dehisced, or the dehiscence happens through a common pore. So, A and B together form one theca, and C and D together form another theca. So that means the microsporangia A and B here, just like the wings, you're finding 2 theca, A and B, and C and D together forming a theca. Remember, A and C, B and D A and C, B and D would not form one theca. They do not share a common pore through which the dehiscence happen, or they are not originating together. A and B originate together forming a theca. C and D originate together forming another theca. Hence, dithecous. Confused? Let's simplify this in our further slides. Now, so far we understood bilobed, we understood tetrasporangiate. Now, there is one ambiguity that is given in the NCERT textbook. So, it is showing that a typical angiospermic anther is bilobed, that's absolutely right, with each lobe having two theca. Now, when you consider or when you read this, you will definitely understand that each lobe is having two theca, isn't it? Now, if that is the case, then there should be four theca, isn't it? If each lobe has got two theca, then it must be four theca. That should be tetrathecous in nature. But that's not the truth, isn't it? Originally, all of them are dithecous only. So when you're reading it, there's a little confusion. So never forget that it is dithecous, not tetrathecous as per NCERT. So always remember bilobed, tetrasporangiate and dithecous in nature. Now let's understand from your diagram which is given in the textbook so that you will clearly understand about the theca. So now what is all forming a theca? So we have microsporangia 1 and 2. A and B, just like how we have seen earlier, and C and D. Now, let's understand that. So, this part is one microsporangia and this is the second microsporangia, right? So, we have two microsporangia and this together is forming a unit which we will call as one theca, because as you can see, they're originating together and the dehiscence happens through this common pore. Likewise, even in the other side also, you can see the same thing here. Now here, so this part, we will consider this part, isn't it? The C and D. Now this part is together forming another theca. So as you can see, they are originated from the same point and they are going to dehisce through this common pore. So we have one theca over here and the other theca over here. Hence dithecous, not tetrathecous. So very clearly bilobed we understood. We understood about tetrasporangiate and also dithecous. Now let us simplify this theca thing once more. So as you can see here, this is our immature microsporangium which you are finding here, isn't it? So this is microsporangium where the pollen is not yet formed, right? Now here, this is acting as pollen sac. Now as you can see, these are layers which we will discuss in some time. Now these layers are not present over here, isn't it? You're seeing that there are pollen grains that are fully formed in each of these pollen sacs. So this was our A, this is B. Again, in A and also in B, we have got what? We have got the pollen grains that are fully developed. And also, you are seeing that the dehiscence is happening through this one common pore, isn't it? So, the pore is common. Through this common pore, you are seeing the dehiscence is happening. So, now after maturing, what is happening? You are seeing that this is forming a single chamber and the dehiscence is happening through this one common pore. So now it is very clear that there are two lobes and two theca, and how the theca are formed, I hope it is clearly understood now. Now let us understand further about the structure of the anther. What are all the layers present? Now coming to the outermost layer, you will find the epidermis. So understanding the whole diagram as you have seen previously, it is really difficult. So we are magnifying this particular part and we are able to see everything clearly and we will understand also everything clearly. So the outermost part is the epidermis, which is a protective layer. Beneath epidermis, you find a single layer of endothecium, and I hope you're all finding these white fibrous structures, isn't it? So these are called as the alpha cellulose fibers, which are very important for dehiscence. So endothecium with these fibers is able to detect the favorable conditions outside, and that is when they break open so that the pollen grains can be released outside. So that's the importance of endothecium. Now we have middle layer beneath this. Usually there are 1 to 3 layers of middle layer, and then we have tapetum, which is a nourishing tissue. So this is the nourishing tissue for the sporogenous tissue which is present inside the chamber. This is the factory where the pollen is going to be formed. So for that purpose, for the pollen to be developed, it needs proteins, isn't it, more lipids. So for that to be synthesized, tapetum is polynucleate in nature. You can find multiple nuclei or sometimes even larger nucleus, isn't it? So this is helpful for the nourishment to be done properly to the sporogenous tissue present inside. And we have connective over here, which is nothing but the joining tissue which is present in between the microsporangia, and also you have got the vascular bundles present here. Now, as I always tell you, at NEET Simplified, you get beautiful images, the real images under the microscope. Now here, let's go and find out how a typical angiospermic anther, which you used to only see in your diagrams or in the textbook, how will you find them under the microscope? We'll meet again after that. So what you have just seen is the diagram which you find in your textbook. Also, you have just seen the exclusive hand-drawn diagram from us as well. So we have understood so much from the pictures. What are we bringing here is the live image under the microscope of a lily anther. Now, why are we doing that? Just because pictures speak lot more than words. Learning from pictures is much better than learning from textbook. So what are we seeing here is the cross section of a lily anther which we have done and fixed it on the slide. Now this is the same slide which we have magnified for you and showing you the exact image under the microscope of what you have in your textbook. So yes, you are seeing the image of the anther. Now, as I have just told you, it is roughly resembling the butterfly, isn't it? Now, this is a typical angiospermic anther. Now, what are we seeing here? You are seeing 1, 2, 3, 4. 4 microsporangia, and this part of the anther forming first lobe. This is the second lobe. This part is forming the second lobe. Hence, bilobed. 4 microsporangia you are seeing. And now let me tell you, this yellow part inside is nothing but what? You have got the pollen grains that are completely formed. Now that is the yellow part which you are seeing here. Now, after the formation of the pollen, we are not going to call these as microsporangia. Instead, we are calling them as pollen sac. So we have got 1, 2, 3 and 4 pollen sacs. Now after the formation of pollen, what's happening is this part is forming a single chamber and this is forming another chamber. Now let me also tell you, because of the only reason that this and this pollen sacs are curled inwards, you're not able to find a clear chamber, but instead you always find one chamber over here and the other over here, hence dithecous. So very clearly we have got bilobed tetrasporangiate dithecous angiospermic anther is what you're seeing here. And this all white part which you are seeing, which is connecting all the parts of the anther, is called as the connective, and you have got the vascular bundles present over here. It is evident that pollen is formed, pollen sacs are formed. So as the pollen is already formed, you are seeing the pollen sacs here. Now, what are the layers which are absent here? The middle layer and the tapetum are absent. Now, we have got epidermis and endothecium. Now, in a moment, let us show you what is endothecium and how is it looking like, and also a clear image of a pollen grain. So now as you can see here, the outer yellowish layer which you are finding here is the epidermis. Now beneath that you have got endothecium, and inside the pollen sac you are seeing the fully matured pollen grains throughout in the pollen sac. So let's begin with the sporogenous tissue which is diploid in nature. This is going to be transformed into microspore mother cell, or it is also called as pollen mother cell, which is also diploid in nature. Now how will we understand the whole tissue. So let's consider one cell out of it. So let's consider one microspore mother cell. Now this is undergoing meiosis. It undergoes a meiotic division, and you have a microspore tetrad, that is 4 cells. Then we get 4 daughter cells which are each haploid in nature. These 4 microspores are all embedded in a substance called as callose, and this callose degenerates eventually. After that, we have the microspore released out, the haploid microspore released out, and this process so far is called as sporogenesis. Now that's not the end, isn't it? We need the pollen to be formed. So for that purpose, let's understand the pollen development from here. So here, microspore undergoing the first mitotic division. How is that happening? That is a very important thing which happens here. So you have a large vacuole developing in the cell. So this large vacuole pushes the nucleus towards the downside. Now in this stage, what happens? The karyokinesis happens and also the cytokinesis goes on. Now for this cytokinesis, it is a special one because it is asymmetric cytokinesis. This is asymmetric cytokinesis. Now as a result of this, what happens is you get a smaller generative cell, and a larger vegetative cell. This larger vegetative cell eventually engulfs the smaller generative cell, leading to the formation of bicellular pollen, which is haploid in stage. Now, here as you can see, this is immature. You can find the sporogenous tissue over here, isn't it? Sporogenous tissue is there. And now, as you can see here, in the bicellular pollen stage, the pollen is all formed. Now, it's called as the pollen sac, from which you are seeing the dehiscence happening through a common pore. And also it is clearly seen that this is all forming a single chamber. Hence, we call this as one theca. Now let us understand about this asymmetric cell division with a very relatable example, and we'll meet once again after the example is done. So here you heard a word called asymmetric cell division. Now I know that is a little complicated. So here let us simplify that as we do it in every episode. Now I have brought you a piece of cake to understand the symmetric and asymmetric cell division both. Before going to asymmetric, let's understand what is a symmetric cell division. So for that purpose, let us consider this piece of cake as the microspore. Right? Now this piece of cake is the microspore and this is the nucleus. So cytoplasm we have got, and also we have got the nucleus. Now what happens in a cell division? We have two stages, isn't it? One is karyokinesis and the other is cytokinesis. So firstly, what happens is karyokinesis, where the nucleus is divided, isn't it? Now what is happening here? We have got this one nucleus which is going to divide, and now we have two nuclei by the end of the cell division. Now after this, what's going to happen? We have got a cytokinesis that happens. So for that purpose, we have the cytokinesis equally happening, so that we have got two daughter cells by the end of the cell division, with each having one nucleus. So we have got two daughter cells. Now this is the symmetric cell division that happens. Now let's understand asymmetric cell division. Now for the asymmetric cell division, we have another piece of cake. Again, this is what we have: the cytoplasm, the cake, and we have got the nucleus. Now coming to the asymmetric cell division happening in the microspore, what's so special here? What is happening here? Before the cell division, as we have discussed earlier, we have got a karyokinesis and a cytokinesis going on, isn't it? But before that, in this microspore, there is a large vacuole which appears in the cytoplasm. Now, this is the large vacuole. What is it doing? It is going to push the nucleus to the corner of the cell, right? So now where we have the nucleus, it is present in one corner. And yes, this is lying over here. The vacuole is there. Yes, and now coming to the nucleus, we have got a karyokinesis going on and a cytokinesis next. So for that, what happens is we have after karyokinesis, we have two nuclei at this one end, the complete end of the cell. Now, as a rule, we have got that after karyokinesis, there is cytokinesis that has to happen between the two daughter nuclei. So now, between the daughter nuclei, we have got the cytokinesis, the asymmetric cytokinesis, which is going to happen. Now, as a result of this, what has happened? We have got one smaller piece of cake, isn't it? And one larger one. So, and very simply, we can relate this to the generative cell, which has formed with which is smaller in size and having a nucleus, and one larger vegetative cell with a large vacuole and the nucleus. And what happens eventually? This vegetative cell is going to engulf the generative cell and become a bicellular pollen. So, with this example, I hope it is very clear about the asymmetric cell division. Now, let's go ahead and understand about the structure of the pollen. Now, you have seen that it was a bicellular pollen stage, and in more than 60% of the angiosperms, you will see the pollen is released in this same stage as you have seen the bicellular pollen stage. And this pollen grain is going to be surrounded or protected by a cell wall which is of two layers. This is the outer exine, which is made up of a substance called as sporopollenin. Now, this is a very tough substance that is very difficult, or it is impossible to be degraded by any external or any internal substance or agent, right? So this is the outer covering, but if at all it is never being disintegrated, how will the materials get exchanged for the reproduction to happen? Isn't it? So for that to happen, it has got some gaps through which the entry and exit of materials can happen for the reproduction to continue. And such pores or such spaces are called as germ pores, and hence this is a discontinuous outer layer in case of the exine. Right? Now coming to the intine, which is the inner layer, this is softer, made up of cellulose and pectin, and it is easily degenerative also. Now coming to the vegetative cell, as you have seen so far, and also the generative cell that you have seen. Now coming to some angiosperms, I mean less than 40% of angiosperms, there is something happening here where this generative cell undergoes mitosis, giving rise to the production of two male gametes, 1 and 2, and we have already have the vegetative cell. Now in this stage, this is called as the tricellular pollen. So that's how we have the tricellular pollen, and this is happening, or the pollen is released in this stage only in less than 40% angiosperms. Now let us understand what are these terms in case of gymnosperms as well. Now in case of angiosperms, we have the stamen. Now equivalent to stamen in gymnosperm, it is called as microsporophyll. In angiosperms, we call anther. In gymnosperms, it's all microsporangia together. And microsporangium is same in angiosperms and gymnosperms. Likewise, sporogenous tissue and also pollen is called as male gametophyte in both of them. Male gametophyte is nothing but male gametophyte, which is nothing but tiny male plant. So that's it for today. I hope we have simplified this complicated concept. Great that you have stick till the end. Keep up your efforts. Stay consistent, stay curious, and I'm here for you till your NEET success. Thank you.